Okay, so this is part two of the presentation on proteins, and we're about to get deeper into enzymes. So still looking at protein structure, or the four levels of making a protein. The very first level, or primary, is simply the connection of amino acids in a line. After this, there are attractions and repulsions between the um, parts of each amino acid, and this can make it curl in and fold on itself in either the form of a helix or a pleated sheet. So those two that you see next to primary are the secondary options or the secondary structure. So we were looking kind of at that pleated sheet version. If certain amino acids were attracted to each other, it would start to fold in like this. Then it can curl in on itself more in a 3D way. So you're connecting peptides and they're kind of sticking out of the page and going backwards in more of a blobby, globular shape. And once you get a number of these tertiary subunits connected together, we call this quaternary structure. And not every protein makes it this far. So not every protein needs to be this complex to do its job. Now there's just a number of different slides that you can look at if you need more instruction on how this works, but I just thought I'd include different options in case my description wasn't easy enough to follow. So you can pause on any of these if they're helpful. So one example of a quaternary globular protein would be hemoglobin. Hopefully that rings a bell. Hemoglobin is the oxygen carrying protein found in your red blood cells, which make up about half of your blood by volume. Hemoglobin has a quaternary structure. So if you follow this chain here, we've got our primary structure, the amino acids connected, and then all of them kind of twisting and folding in due to these attractions here, forming that alpha helix shape. That's the secondary structure. So, yep, it can form either. So you've got the helix there or the pleated sheet. So I'm not sure when it forms one or the other, but that's your secondary structure. When these start folding in more of a 3D globular shape, you're creating this blob here. This is the tertiary or third kind of step in protein structure. And then 4 of these subunits, these polypeptide subunits, come together to form a really large molecule called hemoglobin. So there's 4 of those there, and that has a quaternary structure. So made of many alpha helices, hydrogen bonds stabilizing, all of these helical sections inside the protein. So creating a protein of a specific shape, really. So hemoglobin, this molecule here, makes up about 96% of your red blood cells by dry mass. Each hemoglobin molecule can carry 4 oxygen molecules. So each subunit that you can see here has a heme, which has a ion, as an IRON, ION, which is the unit that actually binds to the oxygen when the red blood cell is passing through the lungs. And hemoglobin molecules can actually bind to carbon dioxide as well. So they carry about 25% of the carbon dioxide back to the lungs. The rest is just dissolved in the plasma. So hemoglobin can actually connect or bind to many different gases in different animals. So we use it to transport oxygen. Okay, and it's not easy to recognize how small this is and how important. So each red blood cell, which you can't even see with the naked eye, you need to use a microscope to see something this small, contains around 250 million of these hemoglobin molecules. So this is just a more detailed version of this here, showing the helix structure rather than just these tubes. Okay, which means if each of these molecules can carry 4 oxygen molecules, connected to its 4 ions, that's the opportunity for each red blood cell to carry 1 billion oxygen molecules. Okay, and then you've got millions of these in your blood at any one moment doing that job. So really impressive and essential protein for us. So that's just one example of one type of protein. I believe that would be a transport, sorry, it's hidden by the taskbar here. Transport protein, maybe a storage protein. I'm not sure, but here are just a few of the main types of protein. So we have proteins that are responsible for structure in the body, like collagen in your skin, which keeps your skin nice and tight and depletes over time, so you get all wrinkly. Keratin in your hair and your nails, keeping them nice and strong. So that's a protein whose job it is to maintain structure. You've got proteins that are involved in motility or movement, such as actin and myosin, which are the proteins that make up your muscles. So you can see it over here. And action potentials, or those nerve impulses, tell the muscles to expand and contract so that you can move. So those are another type of protein. You've got regulatory proteins, which switch your genes on and off. So there are certain cells in your body that do jobs that other cells wouldn't do. And so genes are switched on and off in those cells so that they can all, do their jobs. For example, one of the jobs of the liver is to remove toxins like alcohol from your blood. And so to do this, liver cells, they express genes that encode pieces of an enzyme called, I think, alcohol dehydrogenase. So that's the enzyme that breaks down alcohol. But the neurons in your brain don't remove toxins from the body, so they keep the genes that code for that enzyme switched off. Similarly, the cells of the liver don't send signals like your neurons do, so they keep the neurotransmitter genes switched off. So different cells with different jobs have different genes switched on and off so that those aren't being readed and encoded by ribosomes into proteins. And it's protein's job to do that in each cell. You've got storage proteins like casein and ovalbumin, which are found in milk and eggs, so they just store high energy macromolecules like fats, lipids, carbohydrates, things like that. You've got hormonal proteins. So hormones, most hormones are proteins, and that includes things like insulin, which is a hormone that breaks down the sugar that you eat, and adrenaline, which is a hormone that stimulates heart rate and constricts your blood vessels so that you get a high blood pressure and you can pump blood through your body and get the energy you need to run away from that bear. You've got proteins that are receptors. So they stick out of the plasma membrane so that they can receive information. And the one that we are focusing on is a type of protein called an enzyme. And that is the focus of the rest of this screencast and perhaps the next one, because I see that we are 8 minutes through. So enzymes help the body carry out all of their chemical reactions. Okay, so we'll move on and have a look at that. So I need you to write this down. It's really essential that we fully understand what a protein is and how it works. An enzyme. So an enzyme is a type of protein. So proteins being the larger group. An enzyme is just one type of protein, as we saw on the last slide. And an enzyme is produced by a living organism, and it acts as what we call a biological catalyst. Now, biological is a word we use for anything that is living, bio meaning alive. And a catalyst, hopefully that rings a bell for you from last year. A catalyst is something you can add to a chemical reaction to speed it up or to get it started if it doesn't have enough energy. So enzymes are living catalysts. They speed up chemical reactions in the body, but they don't get used up. So they're not a reactant going into the process. For example, I like to think of it as if you're making a cake, all of the reactants are the ingredients, like flour and eggs and sugar and things. They're going to get used up and turned into the product or the cake. The catalyst, in this case the enzyme, would be the oven. So you have all the ingredients to make the cake, but it can't make itself until you heat it up. Okay, but the oven doesn't get used up in making the cake. You can use the oven again and again and again, but the cake couldn't get made without the oven. Does that make sense? So an enzyme allows a chemical reaction to take place, but won't get used up. And it isn't part of the reaction. It's not one of the ingredients, and it's not involved in the product or the cake itself in the end. In any living cell, thousands of chemical reactions are occurring at the same time every second. So I've got an equation here that says there's 10 to the power of 9 reactions per cell per second. So if you times this by the number of cells in the body, we actually have 37,000 billion chemical reactions. So that's 37 with 21 zeros after it, occurring per second in the human body. So throughout all of your cells. 37,000 billion. Okay, so almost all of those reactions involve enzymes that allow them to occur, so that all of these reactions can take place. And these reactions collectively are known as metabolism. So your metabolism is your body's ability to carry out chemical reactions that break things down and build things up every second. All right, so you might remember enzymes as looking something like this. An enzyme, used to be thought of as kind of like a Pac-Man, so it's got like a little mouth there. It takes things in, chomps them up. We've got two different types of enzymes. Some put two reactants together and build them into a new product. So you can see that on the green one. And some enzymes take in a product and split it into two. So that yellow one's supposed to be a GIF that works. Let's see if I can make it work. Cool. So every time I go to screencast, that's when the issues arise. Well, you can imagine the opposite of the green. So taking in a larger molecule and breaking it into two. So an enzyme is a type of protein, and it has what we call an active site. So that's like the mouth that you see there. The active site is where the actual reaction takes place. The substrate is whatever it is that enzyme is used to process. So each enzyme is specific for one type of chemical reaction. So that green one will only take in blue circles and red squares and join them together. That's its only job. So the active site is specific to exactly what chemical reaction that one enzyme carries out. When the enzyme and the substrate are binded together, so as you can see they connect, that's called an enzyme substrate complex. And what actually happens is that as those two substrates come into the enzyme, the active site changes its shape slightly to kind of grab onto the substrate. So it's not a perfect lock and key. It kind of moves and morphs around the substrate as they bind together, and we call that induced fit. So induced is to make something happen, so it makes itself fit around the substrate. So those are your keywords there. You need to get used to using those when describing enzymes. Substrate, active site, enzyme substrate complex, and the fact that when they're stuck together, this is an induced fit. Okay, so on the right there, you see kind of what we were looking at on the last slide, which is the very basic, artist's impression of an enzyme, but it's a lot more complicated than that. So on the left, you can see kind of the larger molecule itself. So all the different colored blobs are different atoms or molecules that are bound together to form a big, large polypeptide protein that acts as an enzyme. And in this case, this one enzyme works on hydrogen peroxide or H2O2 only, and that will bind to the active site over here and be broken down into whatever it is. Hydrogen, oxygen, water, I'm not sure. Okay, so it's a lot more complex than we make it look. The active site there will morph around that H2O2 molecule in an induced fit and then process it however it needs to. Okay, gotta go.